much for joining us for the public lecture series at Psyche. Psyche is Science Gallery Bengaluru's fifth exhibition season and our third fully online exhibition season. Science Gallery Bengaluru, for those of us who are new here, is a new institution for public engagement with research. We are supported by the government of Karnataka, and we look forward to having you all explore our exhibition season, Psyche, which opens up the complexities of the human mind. Today, as a part of our public lecture series, we are very pleased to have with us Professor Vidita Vaidya from the Tata Institute of Fundamental Research in Mumbai, and she will be delivering a talk titled Serotonergic Psychedelics, a Revival. I'd like to now briefly introduce Vidita, who I've had the pleasure to study with. Vidita received her undergraduate training in life science and biochemistry at St. Xavier's College in Mumbai. She obtained her doctoral degree in neuroscience at Yale University, and after postdoctoral fellowships at the Karolinska Institute and Oxford University, she returned to a faculty position at the Tata Institute of Fundamental Research. She is a fellow of the Indian National Science Academy, the National Academy of Sciences, and the Indian Academy of Sciences. She has received the National Bioscientist Award in 2012 and the Shanti Swarup Bhatnagar Award in Medical Sciences in 2015. Her research group is interested in understanding the neurocircuitry of emotion, its modulation by life experience, and the alterations in emotional neurocircuitry that underlie complex psychiatric disorders like depression. She's committed to mentorship, equity, and diversity in STEM. Before we move on to today's lecture, I'd like to remind the audience about some of the programs that are coming up next weekend. So do watch the film The Task by filmmaker Lee Leder and join us for the film discussion between the filmmaker and social psychologist Yashpal Jogdan on 28th April at 6:30 p.m. And if you're interested in video games, we have an amazing masterclass by Kelly Dunlap, a clinical psychologist and game designer, called Video Games 21st Century Culture and Controversy. So this is on 28th April at 8 p.m. in the night. I would like to remind the audience that there will be an interactive Q&A session at the end of the lecture, so do add your questions in the Q&A box. And also, please do fill out the feedback form. The link will be shared in the chat with you. We would love to know what you think of the talk, but also what you might look for programming from Science Gallery going forward. Now I'd like to hand over to Vidita.
Thanks, Madhu. I'd like to thank the gallery, the Science Gallery Bengaluru, for inviting me to give this talk. So big thanks to Madhushri as well as to Janvi. And I'm going to share my screen. And if you can just confirm that it is indeed visible to everyone. Madhu, can you see it?
Yes.
Hello? Great. So let's dive in, right? I mean, what we're going to do is take a journey back in history to look at our ancestors and to look at hominid civilizations and our lifelong ancestral relationship with psychoactive plants. So as long as humans have walked this planet, we have consumed substances which would alter our mind states. And this has been going on for millennia. Let's look at this from a historical perspective. So if you look at the history of psychedelics, and this is from the Visual Capitalist, and if you just follow along with me, what you'll see is perhaps the earliest recorded history is around 4000 BC, though we know it will be earlier than that. And if you think about the agricultural revolution around about 10,000 BC, remember that we would have been producing barley and wheat. And a lot of the funguses that grow on those grains, in a sense, include the ergot fungus, which produces substances that resemble LSD. So it's probably earlier than that, though when you look at the recorded history, it's around 4000, 7000 BC is what we're talking about. And then over a period of time for several centuries, you see utilization in the Middle America, so in Mesoamerica, where it is used as part of ritualistic ceremonies. And then there is this long period all the way into like around 1500 AD until you have the Spanish Inquisition and the Spanish invasion, in a sense, of Middle America. And you have that period where some of these substances start getting associated with witchcraft. Not surprisingly, given that they produce altered states of consciousness. So some of the compounds that we are going to be talking about are derived from the substances on the right. Peyote is a cactus, and it produces the substance mescaline, which is a potent psychedelic. There are about 100 different kinds of mushrooms that are used, quote unquote, under the terminology magic mushrooms. They produce a substance called psilocybin, which gets processed in our body to form psilocin, which is the active component of these magic mushrooms. And ayahuasca tea, which is used in a variety of Amazonian indigenous tribes, is given as a tea, which is derived from a few different plants that are cooked together to produce this tea. It's a very interesting period of time, and you have centuries of utilization, and really no reports of very severe adverse effects during this period of time. Of course, there are interesting imagery associated with consumption of these foods. In a sense, you can really think of them as foods, or sort of components used with ritualistic spiritual ceremonies, etc. Let's continue to kind of look at this history and take a sort of historical perspective on this. So some of these interesting cave paintings show up around about 4000 BC in Spain. And while the dominant image here is that of a bull, what you will see on the right-hand bottom side is these 13 spindly structures, which remarkably look like the psilocybin mushrooms, okay? So you can actually look at them, and they're lined up almost in a line there. And then actually, when you look at some of these, and this is actually much older than that, and this is supposed to be the mushroom man. This is in Algeria, so North Africa. And what you see is a depiction of what look like psilocybin mushrooms in this medicine man or mushroom man's hands. It's also been debated, and of course, there wasn't evidence for this to say that actually this is indeed a representative of a magic mushroom or a psilocybin containing mushroom. It was more conjecture in a sense. For a long period of time, it was conjecture. What was very interesting is when you start looking at the nature of cave paintings, they often have these fantastical images associated with them, which would not have been part of your standard sensory experience. I mean, you would imagine that that bull is part of the sort of standard sensory experience for people of this time, right? I mean, if you had a little bit of farming and agriculture coming into place, it would not have been surprising to see domestication of animals, etc. But creatures like this, the one on the panel on the right and the creature certainly at the bottom, are not something you would have imagined as part of the standard sensory experience of many of these tribes, communities, etc., that were drawing these interesting cave paintings. And so there's been this debate and discussion about whether there was actually consumption of substances that resulted in an altered state of consciousness. Part of the challenge is actually proving that, which means you need to demonstrate that there is consumption of this nature. And that, as always with all historical records, is difficult to do. There have been some interesting attempts to show it, though. And I actually just want to highlight this example to tell you that it doesn't matter which century you're in. You may have the representation of almost psychedelic-like experience evoked art seen in periods which are several thousands of BC to far more recent, shown here in this image. And what emerges is that the sort of images that are seen and associated with a psychedelic experience involve substantial change within the visual part of the brain, and there are certain hallmark features, and they seem to show up no matter when the person consumes psilocybin. And that's why it becomes interesting to think about whether these cave paintings represented that. But again, let's accept that it's a little bit of conjecture, because we don't actually know whether there was consumption at that time or not, unless we prove it, right? So are there ways to prove it? So this is a very recent paper. This came out in the Proceedings of the National Academy of Sciences in 2020. And this is from what's called a Pinwheel Cave in California. Right now, you can think about this entire region. It's a continuation with the Mesoamericas, right? Which is where the maximum usage of these substances had been reported. In this cave, it's called the Pinwheel Cave because you have this, you can see in the bottom left here, a zoomed up image which looks like a pinwheel. That's why it's called the Pinwheel Cave. And there really wasn't any proof in a sense to say that, you know, altered states of consciousness actually drive the production of these images in caves and rock shelters, even though there is a theory and a hypothesis that ASC, which is altered states of consciousness, were directly associated with the production of some of this kind of art. Problem is, unless you show consumption, you have no way, you're making a guess, right? Now, what's remarkable is when you look at this pinwheel, it really resembles this particular flower of the dhatura plant. And many of you would recognize dhatura because if you have wandered around the streets of, well, many streets in India, you would have seen dhatura. Okay, and it often looks very purple and it is considered to be fairly toxic and people would avoid it. And dhatura is quite interesting because it contains a high concentration of a very interesting substance, which is not a serotonergic psychedelic, but psychedelic-like, and can produce some of these very strong altered states of consciousness. So what was done in this study is they actually went in and tried to find if in this cave there were any leftovers indicating consumption, right? And they found these things that are called dhatura quids, which were stuck within the stone sort of surfaces of this Pinwheel Cave. And then they actually looked at whether it indicated mastication. It's very interesting because you can actually see the shape of these quids and identify chew marks, okay, incisors, molars, etc. And so that was looked at. And then they carried on further with that study and actually did LCMS, which is essentially asking, by the way, they first carbon dated it. So we know that it's like 1300 AD. So we have some idea of when this consumption would have been. It's 1300 AD. And now they're taking that stuff that looks like it's been chewed and then stuck to the walls of this cave and saying, what's inside this, right? And the beauty and the power of the scientific tools of today is that you can identify the signatures that are present in these, the molecules that are present in this chewed up piece that is stuck in this wall. And what you can see is there's scopolamine, there's atropine, and these are very interesting molecules. So scopolamine is the molecule that I mentioned, which is not a serotonergic psychedelic. It actually hits the muscarinic receptor, different neurotransmitter family. It's a blocker of the muscarinic receptor, but very interesting molecule, also known to produce altered states of consciousness. So this, in a sense, they consider as unambiguous confirmation of the ingestion of what they call hallucinatory-like substances at a rock art site. And this opens up all kinds of interesting questions about the relationship between consumption of these molecules, the art that is emerged in these rock caves, and the relationship between the two. And also the fact that this is again proof of the use of these substances across diverse cultures across the globe. And this is, of course, the Mesoamericas. And then you look at a variety of other civilizations, the Aztec, the Inca, the Olmec, the Zapotec, and this is all Mesoamerica civilizations, and they are using these serotonergic psychedelics, which can also be called entheogens. And entheogens was considered to be a far more appropriate term in some sense, because these are, entheogens would be defined as a psychoactive substance that changes the state of consciousness and alters the perception. Perception would be how you see the world, see, hear, feel, you know, whatever, acknowledge or percept of the world, your mood states, states of consciousness and awareness of self and other, cognition, which would be associated with your being able to understand something, remember it, learn it, and of course, a variety of behaviors. And you were using these substances to alter these states of consciousness such that you could actually drive spiritual development or in some sort of a sacred ceremony. And that's why they were called entheogens. Entheos coming from full of the god, inspired or possessed, and genesthai coming from coming into being. So you are using this to come into being to a higher level of inspiration, possession, and awareness of some spiritual state, etc. It's quite fascinating when you look at some of these things that were used in ceremonies during that time. And here is an example of a statue of a god within the Aztec civilization, where you can actually look at the god and you see that there are mushroom earrings. The tops of mushrooms are actually depicted as earrings. There are actually examples of the morning glory plant, which is again produces strong psychedelics. And so you have this interesting depiction of the things that you're offering as an offering to this deity in a sense. And here on the right are Maya mushroom stones from Guatemala. And here is an example of priest bearing psilocybin mushrooms to the god. This is again from Mexico. And what this collectively represents is it was almost viewed as flesh of the gods and an offering to something that was higher or larger or out of just the form of the human species. It's very interesting that this is seen across a variety of parts of North and South America. And we'll also discuss how this shows up in Greek civilization. It showed up in Turkey, in China and India. There was, of course, opiates, which were being used in a major way. Opiates were also being used elsewhere. There are a variety of substances even within the Indian traditional medicine system, as well as the Chinese traditional medicine system, that have this kind of a psychoactive state that produces an altered state of consciousness. The shamans, which really represent a number of indigenous tribes of the Amazon, they are the witch doctors or the healers or the ones who are thought to be the conduit to a higher state, used to brew this tea called the Ayahuasca tea, which is cooked from a couple of these substances. Very interesting molecules derived from these plants. And on the bottom is dimethyltryptamine, which is a very potent serotonergic psychedelic, abundantly found in many different plants, also present in your own body. And that opens up very interesting questions there and of itself. And the molecules on the top are things like tetrahydroharmine, harmine, and harmaline. These are molecules that prevent dimethyltryptamine from being broken down very well. So not only do you have this psychedelic, but you also stop the ability to break it down quickly, which means it's allowed to hang around in the body and produce effects for a while. And, in this period of time, and this is now, we're talking about thousands of years ago, routinely being used, there was already an understanding that there is a set and a setting. You had to purify yourself before you consume these substances. There was an awareness of the fact that you could have, quote unquote, a bad trip, which would result in quite scary visions and visual perceptions. And so there was actually an exercise that you had to go through. It was done under controlled circumstances with the presence of an experienced shaman. There were Ikaro songs that were sung in many of these ritualistic practices, and it was associated strongly with the idea of healing. And this opens up interesting questions. So, of course, we're talking about North Africa. We're talking about Spain from the rock paintings that you see in the caves. You're talking about Middle America, Guatemala, Peru, Southern California, Mexico. So widespread usage of these compounds. And here's an interesting insight into the fact that there were also the Greek civilization also played around with some of these very interesting substances. There's the temple of Eleusis, which is thought to be associated with certain mysteries. Aristotle, etc., are thought to have visited. The temple was eventually destroyed. And Brian Morarescu has written a really interesting treatise on what this meant for the origins eventually of Christianity. And it's very interesting to read and think about and discuss. So many of you may already know about Persephone and Demeter. Persephone was taken away by Hades to hell. And there's this entire story about Persephone. But here's an example of Demeter showing barley, etc. These are the Eleusinian mysteries. And there's this entire debate about whether what was on it that caused these altered states of consciousness, which was very much associated with these temples. It turns out that most of these grains, things like barley, rye, wheat, etc., often when stored will allow fungus to grow on them. And all of us who have left bread in our fridge will have found that we can nicely find molds growing on them, right? It's not very surprising. So you can imagine even in a refrigerator, mold will grow out when left in the open. There was mold growing on all these grains that were stored in granaries all the time. And one of the funguses that grows, one of the fungi that grows is ergot. It's an ergot fungus. And ergot is actually the source of a molecule very similar to lysergic acid, LSD, lysergic acid diethylamide. And the fact that 12,000, 13,000 years ago, we were already seeing this from our agricultural usage opens up the possibility that infected grain was consumed and it was associated with visions, right? I mean, it's not a stretch to imagine this. It's certainly been speculated, it's been discussed quite a bit. Then it opens up the idea that what happened after all of this, right? If we were using this for thousands of years, I mean, going all the way back to 12,000, 9,000 years ago, what suddenly happened in the last 500 years? One of the interesting ideas is the spread of religion and the association of power with religion. And what happens when you have the possibility that the church is very frightened at the idea that if you can consume and curate your own spiritual experience, you may not require any conduit to so-called higher powers. And so, yeah, it says, and this is a direct quote from Brian Morarescu's book, where he says that you obviate the requirement of controlled structures like the church or other controlled religious practices. If you can go and find God in a glass of wine or in some barley that you may consume, what do you need the priest and the bishop for, right? So it opens up very interesting debates and ideas. I'd just like to summarize some of this history by looking at the cultural distribution of the use of psychoactive plants and what are they used for. So if you look on the x-axis, it tells you it's a hallucinogen, it's a stimulant. Stimulant would be something like caffeine, for example, anxiolytic, antidepressant, sedative, analgesic, aphrodisiac. All of these derive from psychoactive plants, and they have actually looked at something like more than 126 plants. And what will emerge, and you can see across different civilizations. So here's Australasian, temperate Asian and Indo-Malayan, European, African, Middle East, multicultural and Native American. And what will show up as one of the largest categories is actually the use of hallucinogens, of which most of them are serotonergic hallucinogens. And I haven't defined that term yet, but I shortly will. That's one of the largest consumed substances used extensively across Native American civilizations. All right, so we come back to this image from the Visual Capitalist, where they are describing the history of psychedelic usage. And we've gone all the way up to this window in which Europe starts, you have movement of people from here to Mexico, to North America. This is just before that window in which colonial spread is happening across the globe, right? And it becomes quite interesting in this period because this is when peyote starts being described in a sense as witchcraft. And we'll talk about that because this becomes quite interesting because this window will rapidly move into 100 years of prohibition in a sense. Very rapidly things moved into prohibition. So what happened where for something that was used as part of food for thousands and thousands of years, how did we move as a people from that to being so worried that it got completely banned, right? And we're talking about substances that did not produce the kind of effects that opiates do. Heroin, certainly, for example, and opium-associated death is not associated with these kinds of serotonergic psychedelics. This is not to say that they don't produce some interestingly scary effects. They absolutely do, but they're of a different class of molecules and produce very different effects on the brain as compared to some of the other drugs of abuse, for example, cocaine and heroin. All right, so let's keep going. Let's talk about something called the peyote effect, okay? And this goes back. So now when we start worrying about bans, like when did it go to the point where somebody said, this is just bad, we've got to stop it, right? So our first example of a ban, at least to the best of my understanding from my reading, is the peyote ban. Okay, and now I'll read this to you because there's a little description that is written by Aurelian Boyard, where he talks about how the Spanish encountered peyote soon after they came in to sort of conquer the Aztec civilization, okay? And they were vigorously opposed to the practice, not so much because they were worried about what kind of effects it has on the body. By the way, they realized that the effects were not restricted to just the Aztecs. They also consumed it, they also had effects. And then it got worrisome because it was associated with religious significance for populations that they wanted to convert, okay? And that was one of the big drivers to actually ban this. And it became such that eating peyote cactus got almost as bad to the padres of the Spanish Inquisition, almost as bad as eating human flesh. So along the lines of cannibalism. And the Inquisition eventually resulted in a period where by June 1620, it was banned. And this is actually the text. It's a translation of what was written in the ban. And I thought it would be really interesting for us to read, right? We as a people and as a species have explored something, got worried by it, and then banned it. And the language of the ban is very revealing in many interesting ways. So I'll read it to you. Inasmuch as the use of the herb, and obviously this is written for the people of the city of Mexico and the states of blah, 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 blah. I've cut all that off because it describes to you who this applies to. But here's what they say. They talk about the herb or the root called peyote. And they say that it cannot be used really because they worry that it doesn't really seem to do much and is an act of superstition. It's condemned because it is opposed to the purity and integrity of our holy Catholic faith. And it's certain, and they say it with absolute certainty, that this herb or any other herb that exists cannot possess the virtue or inherent quality of producing all the things that it is claimed to produce, which is visions, mental images, fantasies, hallucinations, all, and which are presumably images that is sent down to you from some divine source. This cannot be the case. It is actually the work of the devil, who's the real author of this vice. And so we ban it, right? So this is the text of the actual ban in 1620, which is probably one of the first bans that came down very hard on the use of serotonergic psychedelics. And this is indeed a serotonergic psychedelic. The molecule mescaline derived from the peyote cactus would be considered a serotonergic psychedelic because it hits pathways that are downstream of the normal neurotransmitter serotonin, which is abundantly present in our brain and in our body. All right. So carrying on, let's come back then to this window that we went from thousands and thousands of years of using these compounds to, in the 1620s, the first big ban that happens associated with colonialism, invasions, etc. I mean, there's a separate history to tell about opiates, which is associated with the movement into the East and what that did in terms of also fueling financial growth of many of the invading sort of colonial powers. So it's a very interesting history from that perspective as well. So let's look at the Prohibition era, okay? And it's really not as though the whole century is prohibition. In the beginning, people were trying to figure out what is in this. So remember, this is a period of time where the dyes industry and chemistry is beginning to thrive. You can actually derive and synthesize compounds. You can extract compounds. You can start identifying structures. And the first thing that happens here is Arthur Hefter isolates mescaline from peyote. And that happens pretty early on, like 1897 or so, around about that time. And then there is something isolated from the iboga plant, which is ibogaine. And so it goes until the big event of the last century, which is when Albert Hofmann, who was a chemist with Sandoz, synthesized LSD from an ergot fungus. Okay, he synthesized it in 1938. He was looking for substances that did quite different things, influence circulation, etc. He didn't do anything with it for several years after that. He tried it on himself in 1942, and we will discuss that because that was perhaps a very monumental event in the usage of psychedelics and the awareness that compounds like this can produce such drastic states of consciousness. Just to tell you what serotonergic psychedelics are, because I've been using that term so far, here is the neurotransmitter serotonin at the top. It's derived from a small amino acid, which is an essential amino acid, which all of us consume, tryptophan. It goes through several enzymatic conversions to form a molecule called 5-hydroxytryptamine, which is also known as serotonin. Drugs that resemble serotonin in terms of the fact that they produce effects at serotonin receptors and can alter states of consciousness are called serotonergic psychedelics. Mescaline derived from the peyote cactus, psilocybin, which gets converted to psilocine from mushrooms, molecules like LSD, which Albert Hofmann synthesized, DOI, which is also a synthesized product, and dimethyltryptamine, which is found in ayahuasca tea and in many other plants and also in your body, right? So you have all these substances, and it's an interesting era. What actually happened? If you look at the timeline of that period of time, what's going on? So I'll just remind you quickly, because I want to give you an overview. All of the substances here, ayahuasca, DMT, mescaline, ibogaine, psilocybin, are serotonergic psychedelics. So is LSD, which is synthesized. MDMA would not fall into the classical pool of being a serotonergic psychedelic, even though it exerts effects on many things associated with the serotonin pathway. It is a drug of abuse. It's also known as ecstasy. Ketamine is anesthetic. It is also a drug of abuse, and it is used at low doses now to treat treatment refractory depression. Again, that does not fall into the category of serotonergic psychedelics, even though it can have a psychedelic-like effect. So the compounds that I am referring to as serotonergic psychedelics require that they mimic the effects of serotonin, in a sense, on the serotonin 2A receptor largely. I'll be talking about that as we go on. All right, so let's look at a history of this. Now we're doing a shortened history. We went through that old history where we were looking at thousands and thousands of years ago where it was being used routinely. Let's come to a much more shorter timeline. This is from a recent review, actually not such a recent review, a while ago, but it summarizes what happened during that period of time. Here is the synthesis that we have, an isolation of mescaline that happens soon in this period. Then there's a synthesis of LSD by Albert Hofmann. And right up here is Arthur Hefter. Here is Hofmann. And Hofmann synthesized LSD in 1938. He didn't consume it till 1943. After he consumed it, he had and he consumed not a very low dose. He thought he was consuming a very low dose. He was making notes in his book. LSD is extremely potent and requires very low concentrations to have robust effects on consciousness. And bicycle day is actually the day when he, asked his assistant to help him get home. He bicycled home. He was already in, the floor was no longer the floor, the ceiling was no longer the ceiling, the door was no longer the door. Multiple things were changing. It was quite a terrifying experience, and he details it in quite a bit of detail as he describes that incident. That day is called the bicycle day, and it is, in the sort of psychedelic aficionado culture, it's considered to be quite a momentous day. This period is interesting, right? So there's all this going on. This is also a period associated with war. Remember that soon after this period, right? 1945 is when you have the atom bomb, etc. By 1958, he has also synthesized psilocin and psilocybin. Very interesting period of time. And this soon follows after that, the 1960s, where you have Vietnam War. And I just want to show you some of these images, right? Drop acid, not bombs. Love, not war. And you would associate with this with the period of the 1960s and an era of time in which there was a strong counterculture which was pushing back against the war machine. Very interesting period in history. Ended in 1970 with a robust ban that came down. Richard Nixon, in a sense, putting all of these compounds on Schedule 1, which makes them drugs of abuse, drugs that cannot be purchased, and you would be actually, it's a criminal activity, right? So this is a really interesting period of time and an interesting time in history. And one of the most, so yeah, just indicates that this is another big ban that happens. Remember the peyote ban in 1620s? This is 1970 now. I love this quote from Michael Pollan's book because he describes something very interesting. I mean, of course, everybody will have their own favorite idea of what were the big discoveries of the last century. And he talks about something very interesting. Midway through the 20th century, two unusual new molecules show up, okay? LSD, which is synthesized, psilocybin, which is derived from this particular mushroom. These are organic compounds. They have a striking family resemblance. They explode upon the West. Okay, in a time, they'll change the course of social, political, and cultural history, as well as the personal histories of millions of people, right? Because they consume them and they really alter states of consciousness. As it happened, these disruptive chemistries, as he talks about them, are also coinciding with another major disruptive chemistry, which is associated with another kind of mushroom, a mushroom cloud, one far more ominous, one far more deadly, one far more likely to cause profound damage to humanity, and one not likely to be banned. It opens up a very interesting question about power, politics, the war machine, us as a species, us as a people and a culture and a civilization, that we banned what was on the left and didn't go after what was on the right. Okay, just in terms of loss of life, impact to humanity. It opens up really interesting questions because one of the things that psychedelics do, what's on the left is perhaps the greatest counteractor to what is on the right. Associated with some of these compounds are very interesting states that happen to the brain. What happens is ego dissolution. It is associated with loss of a sense of separateness from not just others, but also from plants, the universe, and awareness of unity with the universe. You can imagine that national boundaries, which are imaginary lines drawn on land or maps that only exist for our species and are really irrelevant otherwise, may be a little irrelevant for cultures and societies that are consuming molecules that are associated with ego dissolution, where your sense of self and separateness itself is thought to dissolve. Associated with mystical experiences, and Michael Pollan writes in his book where he's describing large number of sort of surveys done with individuals who did take serotonergic psychedelics under fairly controlled circumstances. And when asked to describe those events as monumental experiences along the lines of birth of their first child or the loss of a parent or a loved one, which all of us know are profound experiences that change the way we look at the world and the universe. Associated with these compounds is altered perception, hallucinations, reduction of something called the default mode network, which I'd be really happy to talk to you all about much more in the tutorial that follows. But think about the default mode network as what you do when you're just chilling and hanging out and you're not really doing anything active, right? You're not sitting and, maybe cooking something or stitching something or any task. When you're not associated with the task, the default mode network is quite active. It is really associated with your sense of self and self-rumination, self-thought, self-awareness, self-reflection. In a sense, in a very loose way, you can think about it as a network where your ego resides. Okay, that's a very loose definition, but perhaps we can get away with a fairly loose definition like that. It is the network that dominates under rest. And interestingly, when you do a task, then task positive networks take over and the DMN actually dampens. When the task is on board, you're not really thinking about yourself constantly at that moment, you're associating with the task. But otherwise, this is our ruminative network. It's a network that is known to be altered in conditions like anxiety and depression, self-rumination being a key component of some of these psychiatric disorders. And associated with most serotonergic psychedelics is a reduction in DMN network connectivity and activity. In a sense, it's almost as though you shut down your self-rumination, self-awareness component such that the rest of the world can come in, right? Much more dramatically, rather than being regulated and controlled by the DMN. It's a fascinating set of work that came from Robin Carhart Harris, who really drove this finding of showing that the DMN network is regulated by serotonergic psychedelics. It's not surprising because serotonergic psychedelics exert most of their psychedelic effects through this receptor, the serotonin 2A receptor. It's a receptor which is also called a G protein coupled receptor. Again, a jargon that you may not necessarily want to know, but most of these neurotransmitters act on receptors that allow them to signal inside neurons, right? And also non-neurons, astrocytes and other cells as well. So the main receptor that these serotonergic psychedelics work through is the 5-HT2A receptor. All right, let's keep going. This is from Michael Pollan's short YouTube talk talking about how to change your brain. And why does he have this conductor with the head gone? In a sense, it's like if you had a conductor that's controlling everything that your brain does, to some extent, your DMN is like that, right? It's the conductor, it's in charge. When the DMN is out of the picture, the world opens up because it's like having a conductorless symphony where things can now no longer be as easily controlled. Sensory experiences get heightened. A lot of things are now no longer self-referential and become very, very interesting as a consequence of that. People describe this as the first time a child sees a rainbow. We don't experience a rainbow anymore the same way, but the first time you saw something rather incredible, you re-experience it when this sort of a change happens to the nervous system. All right, keep going, we keep going. So here's our timeline, right? So all this is going on. 1970s when this ban happens. Already after this, there is some amount of research going on. It's not like research stopped completely, but before this, in this window, like in the 40s and 50s, there was active research going on. And once this became a controlled substance, it got dampened down quite a bit for a long period of time. It wasn't until 1988 that we even knew which was the primary receptor targeted by molecules like LSD or by psilocybin. And it slowed things down. How do we know it slowed it down? So on the bottom here are actually just the number of papers published. Okay, so the height of the bar tells you how many papers were published in that year. For dimethyltryptamine, which is the active component of ayahuasca tea, here's 1955, here's 2022. So I've just collated all papers ever published that include the word dimethyltryptamine. It may or may not be directly associated with their psychedelic-like effects, but even if you did this broad search, what you can see is there are peaks of period in which the research was really active, then there are lulls, and then there's a reemergence. When you look at ayahuasca itself, you can see nobody really was studying it. And it's very interesting what has happened to this in terms of patenting of indigenous understanding, indigenous knowledge, what that means in terms of perhaps larger machines and, pharma companies accessing this, what how do we negotiate this indigenous knowledge versus it transmitting into active usage, right? An interesting history from that perspective also. And here's ayahuasca, and you can see the resurgence of interest. Here's psilocybin, and here's LSD. There's a big thrust in this period, then again a lull, and then it's coming back. Here's DOI, the compound we work on in my lab, and you can see that that came up much later. It was synthesized much later, kicked in much later, and of course it's been actively used. Somewhere in this, in 1997 is my first paper with this compound, when I was just a graduate student in Ron Duman's lab. So interesting set of molecules, went through a wave of really driving neuropsychopharmacology interest and then faded away because of such a tight ban. And they have reemerged. They have absolutely reemerged. So we come now to this renaissance period, or the renewal of interest, or a revival of interest. Why is it that these molecules are coming back? They're coming back because they're profoundly interesting. Unlike drugs of abuse, they do not seem to really work on the dependency pathway, which is a different circuit. They don't seem to cause the kind of dependence that you see associated with compounds like cocaine, nicotine, which, by the way, has never really been banned, even, and has been used substantially and can produce a lot of health effects, but has a big industry backing it. So you can see that this set of compounds just went into a shutdown. And it's very interesting to ask, why this and not something like nicotine, for example? Of course, nicotine doesn't change states of consciousness in the same way as these compounds do. Also, it's not a threat to the war machine. So it's also interesting from that perspective, or a threat to very dominant narratives that we as people have wanted to thrust forward, right? And some of these molecules make us uncomfortable in our understanding of consciousness. And in that sense, they are fascinating, but at the same time, a little overwhelming and a bit scary. In this period, so right around about 2017, MDMA, which is not a classical serotonergic psychedelic, but is indeed a psychedelic, was given breakthrough therapy status for things like PTSD because of the profound effect it could have on shifting states associated with trauma. Then you have psilocybin getting a breakthrough therapy status. You have the use in Hopkins had been doing work for quite a period of time, starting already in the 40s and 50s and continuing through even the banned period, where there were very interesting studies done on terminally ill patients with cancer and end-of-life states, and the anxiety and despair associated with that end-of-life state, and how profoundly meaningful consumption of some of these molecules were in terms of relieving that anxiety and despair that was. See S-ketamine, this is not a serotonergic psychedelic, but ketamine actually opened some of this window. You have a period in which multiple companies are coming in. You have the legalization of cannabis in many places, which opens the door. And you have a variety of psychedelics now emerging with a huge interest in legitimizing the therapeutic value of these compounds. Now I'll breeze past this a little bit. What you see here is the emerging interest. There are something like, this is March 2021, something like 285 active ongoing clinical trials. Okay. In interest in being used as a treatment for depression and anxiety, for substance abuse, for pain, sleep disorder, large list, and very interesting set of studies that are emerging currently and will emerge in the next decade. This is just to give you a sense of why psychedelics may be now being studied really more than ever. This is just the US, data from the US, where you look at clinical trials initiated from 2000 to 2019, and you can see the climbing numbers. All right. So we've talked today about very interesting plant-derived psychoactives, the tryptamines, psilocybin, dimethyltryptamine. We didn't talk about 5-methoxy-DMT. We didn't even talk about bufotenine, which is produced in frog skin, toad skin actually. Ergolines like LSD, which are similar to structures coming from ergot funguses, phenylethylamines, DOI, which we use in my lab quite a bit, as well as NBOM, which is also used in the lab, as well as TCB2. These are synthetic compounds, which are potent serotonergic psychedelics, dissociatives and deliriants, which don't fall into the class of serotonergic psychedelics, but can also produce very strong altered states of consciousness, other than ibogaine, which actually is a serotonergic psychedelic. All right, so we go from this to now a little bit of time spent talking to you about what we do with this, right, in the lab. There are multiple levels at which you could study this. You could study this as an anthropologist. You could study it as a historian. You could have studied it from a political interest. You could have studied it if you're a theologist and you're interested in spiritual experiences and human civilization's interaction with the idea of mystical and spiritual experiences. This is an area that is open to so many people. You can read about this, you can investigate it. It's also fascinating to neurobiologists because these molecules give us tools to understand consciousness, which is the holy grail for a neuroscientist. One of the hardest things to study. Certainly in rodents, it may not even be where we go. And there are people obviously studying this in humans, non-human primates. There are imaging studies. There are studies that are behavioral and psychological. There are a variety of level of studies that are going on. I want to give you a sense and a flavor of the sort of stuff we do in my lab. How do we study this in rodents? It's going to be a little hard in rodents to look at altered states of consciousness very easily. Often these are subjective and you can't inquire this of a rodent and ask, what did you experience? It's, I wish we could, but in that Alice in Wonderland world doesn't really exist where we'll actually get a response. So we can ask what these compounds do at a behavioral level. We can ask what these compounds do at a molecular, cellular, and circuit level. And this is the sort of stuff we do in the lab. I'm going to tell you a little bit about what we do with psychedelics and hallucinatory states that are produced by psychedelics. We look at a variety of compounds, DOI being one of them, TCB2, NBOM. These are three potent synthetic serotonergic psychedelics. We're equally interested in compounds that may be emerging from Indian traditional medicine and may have psychoactive-like action. Berberine and mucuna pruriens. Alkaloid fraction is known to produce robust hallucinations. We're interested in indigenous plants that come from our part of the world that may also have this kind of a psychoactive effect. We take these compounds, we're interested in what they do to the brain, to neurons. We can look at this by taking neurons and growing them in a dish. We could grow rat and mouse neurons. We can use a variety of mutant mice to derive neurons that don't have the 5-HT2A receptor or don't have another molecule that we might be interested in understanding whether it's a target of psychedelics. We use this in rats and mice. So not just in neurons in a dish, but directly in rats and mice, either given systemically or directly into the brain. And at a translational level, we are interested in asking what this does in human neurons that are derived from healthy patients or patients of healthy controls or patients who have schizophrenia or bipolar disorder or treatment refractory depression. And our interest is to understand what it does at multiple levels of biological organization, spanning from the very molecular bioenergetic states in just energy in a cell, which is the currency of how a cell stays alive and respires, molecular changes that may happen, cellular and architectural changes that happen in neurons, what that does to networks of neurons and how they communicate with each other, and how that may impact eventually behavior of freely moving, behaving animals, right? The challenge is to be able to ask whether each of these effects have a direct correlation or a causal relationship at each of these levels. That's a challenge for the whole field. I'll give you a sense of some of what we do, and I'll wrap up with just one example. Here's work from a senior graduate student in the lab, Toshali Banerjee. Here's a rat. It's a Sprague Dolly rat that's been in the lab. And here's an example of one rat that's been administered the drug DOI. It's given systemically, so it's just given intraperitoneally, and it's free to move around everywhere within the body, including to the brain. Here's work from Gonzalez Maiso that shows that associated with the administration of many psychedelics, DOI, DOM, DOB, mescaline, LSD, psilocybin, but not non-hallucinogens or non-psychedelics, you get this condition called a head twitch behavior. So common to all serotonergic psychedelics is this thing called a head twitch behavior. It's a proxy behavior which happens only with the psychedelics. And so that's particularly interesting to us. Let me just show you a video. You might catch it. Okay. There we go. That was a head twitch. It was really fast. I would play this video again so you can quickly see it. So here's the animal sitting. And there goes a head twitch. That's a head twitch. It's a very typical, quick radial movement of the head, which you can very easily distinguish. And what's interesting is compounds. So here's Toshali. You'll wonder why she also has cannabis in her hand. I'll tell you about that in a minute. But Toshali, that's actually just a cartoon. Okay, there's no real cannabis in her hand. It's just a cartoon image. But here is what happens with head twitch behavior with DOI and NBOM. You get very robust increases in head twitch behavior. It's a hallmark signature of psychedelics. We know what these compounds do. We know drugs like DOI, which is a psychedelic, binds to the serotonin 2A receptor, activates a cascade of enzymes downstream, and drugs like glyceride also bind this receptor, do not produce hallucinatory effects, and also may influence similar cascades. What we showed a while back, Antara in the lab showed, was that the degree to which it regulates some of this machinery is quite different for a serotonergic psychedelic compared to a non-serotonergic psychedelic. I mean, a non-psychedelic ligand of the serotonin receptor. And this is indicated by high activation in this cartoon versus low activation. Forget what's written below. It just tells you multiple components that are downstream of this pathway are differentially influenced by drugs that can produce hallucinations versus drugs that hit the same receptor but cannot produce hallucinations. So what Toshali was interested in asking is, what are the lipid signatures produced in neurons when you actually activate these receptors with these drugs? And the red indicates the extent to which these molecules are made. What you can see, and this is work done in collaboration with Siddesh Kamath, our collaborator at IISA Pune. This is in vitro. So these are neurons in vitro, either the cell pellet, which contains the neurons, or the media, which is things that are secreted by the neurons. And I just want you to focus your attention on this line, which says 2AG right here, 2-arachidonyl glycerol. It shows up in the brain, really robustly activated, shows up in media, which tells us it's secreted by neurons, and it's also made by the neurons in vitro when these neurons see DOI, but not made when they see glyceride, okay, which does not produce hallucinations. It was very interesting to us because now we have something molecular downstream of these compounds, which is very different when two drugs hit the same receptor, but one produces hallucinations, the other does not produce hallucinations. So along with Siddesh, here's just showing DOI, TCB2, NBOM. So every one of these serotonergic psychedelics produces 2AG. Ignore the rest. Just look at 2AG right here. And that's the compound, right? So you're producing large levels of 2AG. Fascinating, because what's going on? Here you have DOI. Here's this entire cascade of things that happen. Where is 2AG? There's something called DAG, but there's no 2AG, right? So what we were wondering is it possible that DAG is actually being broken down to 2AG through the activation of this particular enzyme? And that is what we were able to show. 2AG is a very interesting molecule. It's an endocannabinoid. It belongs to the endocannabinoid family like anandamide. It's much more potent than anandamide. And 2AG hits the CB1 receptor, which is another G protein coupled receptor, which is the target of cannabis. Okay, tetrahydrocannabinol is a drug that stimulates this particular receptor. Turns out that serotonergic psychedelics seem to activate this enzyme, causing DAG to become 2AG, and 2AG stimulates this receptor. We're now exploring exactly what this means for the behaviors that emerge. What does this mean functionally for these networks, etc. I am going to just skip through this part and quickly tell you that it doesn't happen just in rat and mouse neurons. It happens in human neurons too. Here's our work in collaboration with Biju, who's a psychiatrist at NIMHANS. These are neurons derived from healthy controls. They express the serotonin 2A receptor. When we grow these neurons in vitro and expose them to DOI, you get a robust increase in 2AG. So rat neurons, mouse neurons, human neurons give you this big impact on 2AG. Okay. Obviously, we want to ask if this would be different in neurons derived from schizophrenic or bipolar disorder patients, especially those that are prone to perceptual and altered states of consciousness, right? Okay, I'm in the interest of time going to skip this part because, yeah, and just acknowledge the people who've done the work in my lab. I've only touched upon it slightly. We work on signaling pathways downstream of hallucinogens and non-hallucinogens that hit the serotonin 2A receptor. Our collaborators on this are Siddesh Kamath, Ritika Sood, Lekhan Shukla, and Biju Vishwanathan at NIMHANS. I didn't tell you anything about what we do with anxiety-like behavior, but we have a large collaboration with Rishikesh Narayanan at IISc and Alex Kwan at Yale, looking at the circuits in which these drugs actually reduce anxiety. I didn't again get to tell you about work from Sashena, but we're interested in mitochondrial bioenergetics, and we're interested in plasticity, like changes that happen in the brain. Collaborations with Ullas and Ashok Vaidya. And some of this work is already published. It's also on our website. I'm happy to discuss it further. These are the people in the lab who've done the work over the years. And my collaborators, Siddhesh and Biju, who I talked about in terms of the work that I described today. And I think I am done and can take questions. I hope I've kept within time.
Yes, absolutely. Thank you so much, Vidita. We do have time to take questions, so I'm going to, encourage all the people in the audience to share your questions in the Q&A box. I can already see questions are coming up. So shall we just, maybe we can just dive right into the questions from the audience. And also just a reminder that we have a tutorial session with Vidita after the lecture at 8 p.m. So please feel free to sign up and come there as well to continue the discussion, since we don't have too much time over here. So Vidita, I'm going to start off with the first question, which is from Tejas, which is, what is the potential of serotonergic psychedelics becoming addictive to the host? You did mention that the pathway of dependence or compulsion is different, but is there still no risk of addictive behavior arising from regular use of these drugs?
It doesn't appear to be that this pathway, sort of the cortical pathways are the ones that really have an abundance of the 5-HT2A receptor, whereas the ventral tegmental area to the nucleus accumbens is less abundant in the 2A receptor. It doesn't seem to hijack the reward pathway in any substantial extent. Also, many of these compounds lose their effects with repeated use. So, it doesn't have that classical rewarding like element. I think the sort of scary part of these compounds is that they can produce very potent changes in consciousness and perception, some of which can be quite, and people describe bad acid trips or bad trips, which are associated with quite, unpleasant psychedelic-like states. And those seem to be the bigger concerns. Obviously, there would be a concern that you would not want someone, operating heavy machinery or a car or something under the influence. So obviously, but in terms of controlled use, where it's given clinically in the right set and setting, they appear to be very potent molecules and for therapeutic use, and do not have the kind of abuse potential you see with something like nicotine, for example, certainly with cocaine and heroin. Does not fall into that class. And that is true also of marijuana to some extent.
I think the next question also stems from something I wanted to ask you as well, was we spoke a lot about, especially when you talk about the recent renaissance of psychedelics, it seemed to me a lot of it was focused on the sort of Western world. I was wondering what is, like, from your understanding, like, what is the current situation in terms of India? And, so one question from the audience is, do you have problems securing these chemicals for lab use? Is research into these compounds now, sort of happening in full force in India as well? And yeah,
it's not happening in full force in India because it's very difficult to access Schedule 1 drugs. The NIDA, which is the National Institute for Drug Abuse, will actually provide these chemicals for researcher use if you have a license, which requires a license from the Narcotics Bureau. It's not easy in India to get that permission. DOI, TCB2, and NBOM are not Schedule 1 drugs. They're used only as experimental 5-HT2A receptor agonists. My lab has been working with them for the last 22 years. So you see in our work, you will not see work that shows sort of data from LSD and psilocybin. We have sort of cleaner serotonergic agonists that we use. But yes, this is a big challenge. And that's really holding us back within India in terms of really going forward with this. Plus, as far as I understand, there aren't any major clinical trials underway within India itself to see the local usage and relevance to, psychiatric treatment within India. Biju would be much better placed to answer this question than me. But I believe it will happen. Certainly, ketamine went from not being considered to being used, right, very quickly. So I think the way things are changing and the speed at which they're changing, that will happen. We've been very lucky to collaborate with the NIMHANS team because they have a narcotics drug license, and they have been able to sequester some psilocybin for experiments with human neurons. So we will be able to use it in Bangalore at NIMHANS. But yeah, in my lab, this is a perpetual challenge. It is absolutely a challenge, and it really slows us down.
I think this really flows into the next question where, I mean, you already sort of brought it up slightly, that newer forms of treatments that use psychedelics. The question from the visitor is that how can people suffering from a lot of these mental health problems access such treatment? Would such access at all be possible in India?
So currently, these are all Schedule 1 drugs. So these are drugs of abuse in India. So absolutely not. It would be illegal to access them, right? So they are illegal. And the only scenario in which I imagine access will be once psychiatrists open up to the possibility of the use, it gets scheduled for that use, and it is done under controlled setting. I would not, I mean, I want to overemphasize this actually. The set and setting seems to be absolutely critical to the nature of experience, and that seems to really matter. So think about it as if your default mode network goes down, what is available to you for you to sensorily experience is dependent on the direct environment around you. So it does matter. So setting is a big player. Your expectation of what you expect from it is also a big player, right? So anticipation of what you think you're going to experience is also a critical player. So I would say that I don't see this happening right away, but I would expect in the next decade, given the speed at which you're seeing this happening in the West, you're seeing it happening in Australia also, I can't imagine that India will be far behind. Ketamine is certainly already used in India. So that already tells you that has happened. So it's a matter of time, I really think, more than anything else. But currently, it isn't accessible. And what will be interesting to see, Madhu, is that the sort of more experimental stuff, which is asking whether you need the hallucinatory component. Can you take molecules that do not give you the hallucinations, yet tackle the same circuits? And there's already some work that's happening along these lines. So David Olson's group has synthesized something called tabernaclog from the iboga plant, from ibogaine. And that doesn't seem to produce the strong hallucinatory states, but produces some of the mood-related behavioral improvement. So you will see a bunch of things emerging over a period of time, which may not have some of the risks associated with psychedelic exposure. Keep in mind that you can have things like HPPD that arises with usage of LSD as well, where you can have sort of a resurgence of a bad trip decades later without any drug being on board, right? So because these are very potent molecules and they can really change circuits in the brain. So one is not dealing with something here that is mild by any stretch of imagination, right? So yeah, there is a cautionary note to play here. Absolutely. And that's why the setting and clinically controlled usage is critical.
Okay, I think somehow you have managed to answer quite a few questions in the chat, which I haven't yet said, but I'm going to, I mean, the last question that just came in is there a possibility of endogenous 2-AG and anandamide going haywire by some means, and an individual having a psychedelic-like experience without actually being exposed to psychedelics?
I mean, it's a really interesting question. I can't imagine why you might not see something like that, which is that you, without having the receptor, you activate the downstream machinery. Is that in the right population of neurons? Is that sufficient to give you the experience without having the drug on board? I think it is possible to do that. And that opens up a very natural next question, which is what, without the drugs, do these experiences also happen? Clearly they do, right? So transcendental meditation has been suggested. I mean, people talk about experiences of altered states of consciousness, but they're not reproducible very easily. This is the big thing with the molecule giving you the state versus induced states. But induced states also happen, which are independent of the drugs on board. It's not unheard of. So I mean, obviously patients who have schizophrenia have altered perceptual states in the absence of consumption of the drug, right? So clearly the networks that give you those states are there.
Yeah. I mean, because you spoke about the DMN, I just, like, before I go to the next question, I just want to say that we have an exhibit on the DMN and schizophrenia in the exhibition. So for those who are interested, that is from work in NIMHANS by Dr. Urvaksh Mehta. So you can definitely check that out. I think because we're a little tight on time, I'm going to ask just maybe one more question. does the consistent use of psychedelics reduce its effects on the brain and body? Like, the same way, say, do you get, uh, I forget what the word is, like,
You
does it
desensitize.
normal? Yeah,
Yeah, you do.
desensitize.
You do. Absolutely. I mean, you see this in rodents also, which is repeated usage does change the nature of molecular, cellular, structural changes in the brain as well. So it's not surprising given these pathways can desensitize and downregulate. And that's also been reported even in clinical use as well.
So because we are going to take the break, I'm going to again encourage, there are lots of questions in the chat. I am going to encourage those who would still like to talk to Vidita and discuss the questions on their mind. Do sign up for the tutorial. My team will be sharing the link in the chat so that you'll be able to join in for the tutorial. There are so many more things I want to ask you, Vidita, about microdosing. I love the Alice in Wonderland reference. It just reminded me of eating the different parts of the mushroom. It was a really fascinating talk, and I think people are very excited, and there's a lot of interest to know more. I think it's like you said, the fact that sometimes when you ban things and you make things, you sort of put them away, it also increases our interest to want to know more and understand them better. So thank you so much for this really fascinating talk. It was absolutely wonderful. And I just want to let everyone know, if you missed out on any of our lectures or you want to listen to this lecture again, the recording will be up on our YouTube channel. So do check it out. If you've enjoyed this lecture, there is a live lab walkthrough with Anjal from Upinder Bhala's lab, who will be showing some work that they are doing with mice as well. And Biju, who Vidita has spoken about so much today, he and the entire team from the ADBS Nimhans will be doing a walkthrough of their experimental work as well in the exhibition. And yeah, do watch a recording of SP Arun's lecture, The Incredible Perception Lab Inside Your Head, which talks a lot about how we perceive things, how we perceive the world around us, which would be an interesting follow-up from here today as well. Also, because Vidita spoke about Michael Pollan, do explore the Psyche Media Lounge, which contains books, podcasts, and games around psychedelics. And Michael Pollan's work is in there if you're interested. And yeah, I would like to remind everyone to fill in the feedback form, register for our programs, and to see the exhibition before it closes on the 15th of May. Thank you so much, Vidita, for joining us. It was a fascinating session.
Thanks.
And look forward to seeing you all in the next set of programs. Have a good Sunday night, everyone. Bye-bye.